I need a notebook. The next time I go out, I'm gonna get notebooks from my mom. Okay. Okay, I decided I'm just gonna use loose leaf paper because I'm not sure if I'll even want to keep these notes. I think I'm just writing it down for the sake of learning. Chia seeds look like little dragon eggs. This thing made me really so sad. So sad. I want to have like pretty notes, but then like that's an extra piece of like effort. And I don't think I should be going after it. Though I'd like to get my handwriting looking nice again.
So and in structural biology too, there's a lot of experimentally determined structures that are computational virologists can use. So examples of this are advances in high throughput sequence virology. So here are some examples. There's also this, of course. So basically it means the application of computational biology to the study of viruses. So it can be things like assessing the sequence diversity and phylogeny of a virus, modeling the structure of a viral protein, modeling interactions between viral proteins, and modeling antibody recognition of viruses, and then computational virtual screening, so screening small molecules against a viral protein to identify or optimize a viral inhibitor. to get a new drug that can target the virus. And then another modeling thing that people might try to do is modeling glycans in a lot of viruses by constellation on the surface of these viral glycoproteins, and those are often not found in the experimentally determined structures, so people might use modeling to predict how the glycans might look and behave on those proteins. Right now is a very exciting time for computational virologists. So there's a lot of things going on in the field that provide data and information that computational virologists can use. So examples of this are advances in high throughput sequencing. So people can look at things like viral evolution, viral escape, people that are infected with the virus. People can rapidly obtain the sequences of antibodies and T-cell receptors from individuals who have cleared viruses. They can look at repertoire scale sequences of antibodies and T-cell receptors, so millions and millions of these molecules that from the immune system that are capable of targeting viruses. And so that, and in structural biology too, there's a lot of experimentally determined structures that are being solved right now through X-ray crystallography, NMR, nuclear magnetic resonance, and also cryo-EM is a really up-and-coming area that they're creating lots of new structures of viral proteins and large protein assemblies from that. So it's really cool. We have a lot going on in terms of structures. And then, of course, computing capabilities are advancing. These large clusters of computational methods to these various really nice data sets that are coming out. And of course, right now, as everybody knows, is COVID-19. So there's some methods to really great modeling and design programs that we can use to apply computational methods to these various really nice data sets that are coming out. And of course, right now, as everybody knows, is COVID-19. Computational methods to these various really nice data sets that are coming out. And of course, right now, as everybody knows, is COVID-19. And of course, right now, as everybody nice data sets that are coming out. And of course, right now, these really nice data sets that are coming out. Computational methods to these various really nice data sets that are coming out. Computational methods to these various really nice data sets that are coming out. Computational methods to these various really nice data sets that are coming out. And of course, right now, as everybody knows, is COVID-19. Computational methods to these various really nice data sets that are coming out. And of course, right now, as everybody knows, is COVID-19. So this is a viral pandemic. This virus called SARS-CoV-2. And we've actually had a lot of really cool things going on already in the computational biology community in terms of folks being mobilized to combat this virus. And I'll talk more about this in the next few weeks during this course. But people are doing things like modeling the structures of this virus, modeling the dynamic, the behavior of these viral proteins, and also the interactions of the proteins. And other people are looking at rational design methods to design and perform virtual screening to identify compounds that can target the virus. And they're also looking at antibodies and protein-based inhibitors as well. And then also on the protein design side, people are using structure-based design to design their vaccines. And so over here on the right-hand part of the slide, I'm showing this Rosetta community, this fairly recent press release. They're talking about what those people are doing to fight this coronavirus. So one figure here is this de novo mini protein finder. So it is a small protein that the people have designed in Rosetta to target this particular protein on the SARS coronavirus spike. And then also these other teams have looked at ways to assemble these key proteins on the virus on what they call a nanoparticle, and this can be used as a vaccine. So they're doing a lot of cool things in the computational biology community. People are getting together and having meetings and really doing awesome stuff. So this is very it's a challenge, of course, for everybody, but I think a lot of people are stepping up to meet the challenge of the scientific community. Computational biologists are no exception. And so, as I mentioned, I'll fit a little mini protein binder. So it is a small protein that people have designed in Rosetta 2. Really cool things going on already in the computational biology community in terms of folks being mobilized to combat this virus. And I'll talk more about this in the next few weeks during this course. But people are doing things like modeling the structures of this virus, modeling the dynamic, the behavior of these viral proteins, and also the interactions of the proteins. And other people are looking at natural design methods to design and perform virtual screening to identify compounds that can target the virus. And they're also looking at antibodies and protein-based inhibitors as well. And then also on the protein design side, people are using structure-based design to design better vaccines. And so over here on the right-hand part of the slide, I'm showing this Rosetta community, this fairly recent press release. They're talking about what those people are doing to fight this coronavirus. And so one figure here is this de novo mini protein binder. So it is a small protein that people have designed in Rosetta to target this particular protein on the SARS coronavirus spike. And then also, these other teams have looked at ways to assemble these key proteins on the virus on what they call a nanoparticle, and this can be used as a vaccine. So they're doing a lot of cool things in the computational biology community. People are getting together and having meetings and really doing awesome stuff. So this is very it's a challenge, of course, for everybody, but I think a lot of people are stepping up to meet the challenge of the scientific community. Computational biologists are no exception. And so, as I mentioned, I'll integrate this, of course, this current thing into the course. And so we'll have a chance to look at this, both of the slides and also the hands-on exercises, at least some of them. I'll integrate this, of course, this current thing into the course. And so we'll have a chance to look at this, both of the slides and also the hands-on exercises. As I mentioned, I'll integrate this, of course, this current thing into the course. And so we'll have a chance to look at this, both of the slides and also the hands-on exercises, at least some of them. Try to incorporate some of the sequences and structures that are coming out that are directly related to this. All right, so I wanted to give one case study of incorporating some of the sequences that also came out into the course. And so we'll have a chance to look at this, both in the slides and also the hands-on exercises, at least some of them. Try to incorporate some of the sequences and structures that are coming out that are directly related to this. All right, so I wanted to give one case study of incorporating some of the sequences and structures that are coming out that are directly related to this. All right, so I wanted to give one case related to this. All right, so I wanted to give directly related to this. Structures that are coming out that are directly related to this. All right, so I wanted to give one case study of computational virology. So in this case, we're talking about influenza. And so this actually came out, this paper a few years ago in the journal Science. So in 2009, there was an antibody called CR6261. So in the figure on the left here, that's down here at the bottom, this structure of the protein called hemoglobin for influenza. So this antibody CR6261 is what they call a broadly neutralizing antibody. So influenza, as people probably know, varies a lot in terms of its sequence. And it's relatively hard to find antibodies that can simultaneously target one version of flu, another version of flu, and these different what we call subtypes of hemoglobin, which represent sequence, in some cases, structurally divergent forms of the hemoglobin protein. But this CR6261 is a special antibody, especially at the time. Many of these have been identified that way. And so this is a really big paper in Science, and they found it actually down to a different spot than most other antibodies bind. So these on the left here bind up to the hemoglobin head. But this one, CR6261, binds to this key region down the hemoglobin stem. There seems to be a conserved site, which probably explains why it's able to bind these different versions of flu. This key region down the hemoglobin stem. There seems to be a conserved site, which probably explains why it's able to bind these different versions of one line to this key region down the hemoglobin stem. There seems to be a conserved on the left here, and so this is a really big paper in Science, and they found that it actually bound to a different spot than most other antibodies bind. So these on the left here bind up to the hemoglobin head. That this one, CR6261, binds to this key region down the hemoglobin stem. There seems to be a conserved site, which probably explains why it's able to. That this one, CR6261, binds to this key region down the hemoglobin stem. So these on the left here bind up to the hemoglobin head. But this one, CR6261, binds to this key region down the hemoglobin stem. There seems to be. The conserved site, which probably explains why it's able to find these different versions of food, because it's that region that the antibody finds doesn't vary very much. And so these are pretty rare antibodies. There have been a few more that have been identified since 2009 when this came out, but overall, for people that have immune response to it's largely from this one, including the head, because that sticks out. It's a larger area. It seems to be more applicable. So some people in the computational biology community looked at this and thought that you could try to design proteins that target this critical stem region that's targeted by CR6261. So this obviously, this site, as I mentioned, is pretty well conserved. They thought they could try to, rather than using the antibody to target this region, could they use design small proteins to take the place of the antibody, target the site in those completely pan or cross-reactive molecules that can simultaneously bind these different versions of influenza. So this group is part of this developer community, that Rosetta community that I mentioned a couple slides ago. And so David Baker, he's the leader of this Rosetta this group at the University of Washington. Sorrel Fleischman is the first author. They designed several different molecules to try to target this site. So on the next slide is the protocol that they showed in their study. And it actually involves a lot of different steps that I wanted to highlight here because many of them at least pertain to computational techniques and computational virology. So in the first part, of course, they look at the level of conservation, so that entails looking at the sequence and aligning the sequence in what regions of the site that they're targeted by CR6261 and are conserved. Then they looked at structural analysis, so they found different small proteins that might be able to target this site, and then they performed analysis of those structures further to see if they would have what they call scaffolds that would accommodate certain key residues in the context of this binding region. And so these are shown sort of on the left in the middle, about halfway down on this figure. So they have these three critical residues that CR6261 seems to use, and they wanted to see if these proteins could form what they call scaffolds and accommodate these hotspots. And they performed docking simulations with these proteins, with these grafted residues, incorporated the hotspots, and then iterated over the hotspot libraries just to further refine their predictions. Then after that, once they got some good docked models of these scaffolds with these hotspot residues, they performed protein design to further make the proteins better behaved, more soluble, and maybe a better binding to the target. And then finally, Of course, there's experimental work to determine whether these things actually work, which is usually a critical step in any protein design project. And in this case, they did find a couple that did work. I'll talk about that in the next slide. Right, so they screened a large number of designs that they found from their computational pipeline, about 100 or so. And so 90 designs were expressed, but not many of those found hemoglobin. So only 2 out of their 90, there's 3 seem to work, but 2 did. And that's really good still. And one of them is shown here. It's called HB36. So they actually solved the crystal structure of that protein in complex with hemoglobin, which is really nice. So as shown in the figure on the right here, it finds various elements, subtypes in group 1, so H1, H6, H5, so they tested against representative influenza hemoglobins representing those. And so it didn't find several of them in group 1 and also group 2, which is a bit divergent from group 1. But they did achieve a pretty good thing by having this cross-reactive binding protein. And so these designs were both structurally characterized. They tested to see whether they were similar to their models, and indeed they were. So it looks like they have a nice couple of binders, for at least for group 1 HA subtypes. More recent work that this group and others have done have looked at in vivo protection. So they found this thing that binds hemoglobin, that's great. Then they looked further to see whether it actually can protect. So they looked in vivo at these mouse or rodent models. They did find that it actually works and protects these models, these animals from flu, which is great. And also in this review article, they highlight a few other things. So these HB 36.3 and HB 80.4 represent the designs that were discussed by, described by Fleischmann et al. But down on the lower right, people have actually designed them in smaller binders in subsequent studies, including this P7, which I think is really cool. So it's really small, basically just a staple loop that can bind that area with pretty high affinity. So that's really interesting work. All right, so to summarize this, so people in this study, and others now too, Things. So these HB 36.3 and HB 80.4 represent the designs that were described by Dr. Nadal. But down in the lower right, people actually designed even smaller binders in subsequent studies, including this P7, which I think is really cool. So it's really small, basically just a staple loop that can bind that area with pretty high affinity. So that's really interesting work. All right, so to summarize this, so people in this study, and others now too, have shown that docking and design combined together, these two computational approaches can lead to actual effective protein binders. You can use that to target a particular protein and region that you want. And of course, you do need experiments to screen and determine whether these designs that you see on a computer that you generate there do work. But once you do have that capability in place, you can actually figure out which ones work, if any. hopefully characterizing the figure if the structure is matched. The question is, can this sort of approach will work in other systems? You can target other viral surfaces. For instance, hepatitis C virus, which is what my group looks at, and we design protein binders to target that and maybe inhibit viral entry with new novel binding proteins. And so that feeds into the idea of the course project. So basically, that idea here is to do your own version of the study, where you model and design the novel binding protein to target the virus at the site of your choice, the virus of your choice as well. So if you want to attack, for instance, SARS-CoV-2, there's several sites of interest that people have identified there already. You might choose to do that, identify a peptide or a protein, you can dock, design it, even a small molecule if you prefer, learn more about ways to do these sort of things during the course. And so I wanted to relate this anecdotal information and these really cool studies that people have done to the work that we'll be talking about in the context of this project. Well, hello everybody, welcome back. Right now, we're talking about the third part of this first class where we'll discuss databases and molecular visualization tools. Okay, so one database I want to mention to you is called the Protein Data Bank. And so this is a database of experimentally determined molecular structures. So in spite of the name, it's not actually just a protein data bank. It contains structures of RNA, small molecules, other types of molecular structures as well. So its website is shown here, rcsb.org. RCSB stands for Research Collaboratory for Structural Bioinformatics. So the PDB was originally announced in 1971. So I actually dug up the paper that describes that, or announced it here in the journal Nature Biology. So anyway, so that this is early on. I doubt there are very many structures, and certainly far fewer than there are right now. But yeah, so it's been around for quite a while, decades. And what I want to highlight to this crowd is kind of interesting is that the University of Maryland played a role in the development of the protein data bank. And so my institute is now called IBVR, but before that it was called CARB, the Center for Advanced Research and Biotechnology. And, a couple of decades ago, we were one of the organizations that were participating in the PDB, which I think is really cool. So we do have an actual part in the development of the PDB and its history. Okay, so if you go to the PDB website, this is what it looks like. And so the front page, the main page of the PDB has a search box. You can, up near the top, where you can search for the molecule name. There's also an option to do advanced search, sort of look for certain features of the structures or type of structural determination. Is pretty much anything you can think of can be searched for on the PDB through this website. And so the PDB structures, they all have 4-letter code to uniquely identify the structures. It's called PDB code, PDB ID. So it's usually is a number followed by letters and possibly other numbers too. But usually the first is numbers. So 1A, 07 is one of the ones I looked at a lot. 4NWF. And generally over time, other types of molecular structures as well. So this website is shown here, rcsb.org. RCSB stands for Research Collaboratory for Structural Bioinformatics. So the PDB was originally announced in 1971. So I actually dug up the paper that describes that here, journal Nature Biology. So anyway, so that this is early on. I doubt that there are very many structures. Oh, hello everybody, welcome back. And right now we're talking about the third part of this first class where we'll discuss databases and molecular visualization tools. Okay, so one database I want to mention to you is called the Protein Data Bank. And so this is a database of experimentally determined molecular structures. So in spite of the name, it's not actually just a protein data bank. It contains structures of RNA, small molecules, other types of molecular structures as well. So this website is shown here, rcsb.org. RCSB stands for Research Collaboratory for Structural Bioinformatics. And so the PDB was originally announced in 1971. So I actually dug up the paper that describes that, or announced it here, journal Nature Biology. So anyway, so that this is early on. I doubt that there are very many structures, and certainly far fewer than there are right now. But yeah, so it's been around for quite a while, decades. And what I want to highlight to this crowd is kind of interesting is that the University of Maryland played a role in the development of the protein data bank. And, so my institute is now called IBVR, but before that it was called CARB, the Center for Advanced Research and Biotechnology. And a couple of decades ago, we were one of the organizations that were participating in the PDB, which I think is really cool. So we do have an actual part in the development of the PDB's history. Okay, so if you go to the PDB website, this is what it looks like. So the front page, the main page of the PDB has a search box. You can up near the top where you can search for the molecule name. There's also an option to do advanced search for certain features of the structures or type of structural determination is pretty much anything you can think of in certain advanced search. The main page of the PDB's history. Advanced Research and Biotechnology, Protein Data Bank. So my institute is now called IBVR, but before that it was called CARB, the Center for Advanced Research and Biotechnology. And a couple of decades ago, we were one of the organizations that were participating in the PDB, which I think is really cool. So we do have an actual part in the development of the PDB history. Okay, so if you go to the PDB website, this is what it looks like. And So the second page, the main page of the PDB has a search box. You can up near the top where you can search for the molecule name. There's also an option to do advanced search. So for a couple of decades ago, this crowd is kind of interesting. Is that you ever see Maryland played a role in the development of protein data bank? And so my institute is now called IBDR, but before that it was called CAR, the Center for Advanced Research in Biotechnology. And a couple of decades ago, we were one of the organizations that were participating in the PDB, which I think is really cool. So we do have an actual part in the development of the PDB history. Okay, so if you go to the PDB website, this is what it looks like. And this crowd is kind of interesting. Is that you picture biology. So anyway, so that this is early on. I got that there are very many structures, and certainly far fewer than there are right now. But yeah, so it's been around for quite a while, decades. And what I want to highlight to this crowd is kind of interesting. Is that you ever see Maryland played a role in the development of protein data bank? And so my institute is now called IBDR, but before that it was called CAR, the Center for Advanced Research in Biotechnology. And a couple of decades ago, we were one of the organizations that were participating in the PDB, which I think is really cool. So we do have an actual part in the development of PDB history. Okay, so if you go to the PDB website, this is what it looks like. And so the second page, the main page of the PDB has a search box. You can up near the top where you can search for the molecule name. There's also an option to do an advanced search. So for certain features of the structures or type of structural determination is pretty much anything you can think of can be searched for on PDB through this website. And so the PDB structures, they all have 4-letter code to uniquely identify the structures. It's called PDB code, PDB ID. So it usually is a number followed by letters, possibly other numbers too. Usually the first these numbers. So 1A, 07 is one of the ones I've looked at a lot, 4M from that. And generally over time, the first number increases. So a lot of ones you see coming up now start with the number 6. Presumably when they run out of PDB codes that start with the number 6, you'll see the 7 come up around. Code or something like that. See. All right, so when you go to a structure page, so if you type in the first front page, you can search for structure 6-MEJ, for instance, which is shown here. So that's the structure of hepatitis C virus E2, so one of the envelope glycoproteins of HCV, complex with a couple of antibodies. And you see certain things on this page when you go to it. The method that was used to solve the structure, so in this case, X-ray crystallography, the resolution of the structure. So sometimes well, every structure they have a resolution recorded, which tells you how well defined the structure is. But we experiment with lower numbers of the resolution, means higher accuracy. So this one is 2.8 angstroms, which is very good accuracy. So there's an inline structure viewer. You can click on where it says 3D view structure in this box here on the left. You get to better viewing window if you like. And it also has a reference to the journal article, so that's down the lower right hand part. So it's micro article in this case is shown. And you can also do other things too. You can look for other structures that have similar sequences. So there's other options on this page you can go to. It's called sequence or something like that. So there's a lot of things you can do from the structure view window. All right. So what about viruses? So of course the PDB has been around for a while. The question is, can we find viral structures? These are interest for virologists in the PDB. And so the PDB has actually quite a few structures containing viral molecules, over 10,000. And there's a total of quite a few total structures, so over 162,000 as of a couple days ago. So there's quite a lot going on in the PDB. And a large, or sizable percentage of structures are using viruses and viral molecules. So there's lots of represented viruses in there: nucleophage, hepatitis C virus, HIV, flu, enteroviruses, SARS, now SARS-CoV-2 is in that category. If you look here, you can see the amount of different viruses that compose that. So HIV is one of the more well-represented ones, as well as influenza, nucleophage. SARS actually has a fair amount, and a lot of them are what's called other. So you do see quite a few different viruses represented, as well as a lot of structures that represent specific viruses that are of interest to people in solving structures, like HIV. All right, so there are a lot more databases and resources out there, and many of you may be familiar with them. One of them is through NCBI and NIH. It's called BLAST. There's also NCBI, its own database too. You can search gen banking, get protein sequences. And BLAST is a tool on the NCBI site that permits you to search rapidly. It's called basic local line search technique. We'll talk about that later in class and search against structures in their large database collected on it to matches your database of your sequence of interest. There's also another database that I use sometimes called UniPro. So this contains annotations, sequences, matches against structures, other features within the protein that you might be interested in, like phosphorylation sites, signal peptides within the protein. So this is a nice site as well. And for viruses, there are actually a fair amount of database and resources out there as well. So this one here at the top is called Viper. That stands for Virus Packaging Resource, and that contains some neat little pages where you can see they're devoted to specific classes of viruses, dengue, Lassa, Zika, Hep C virus. So we have a lot of things represented on there, so they contain sequences, they contain molecular structures. This is a really nice, fairly comprehensive database resource. There's also Los Alamos National Lab. teacher in biology. So anyway, so this is early on. I got Leader for Advanced Research in Biotechnology, the PDB website. This is what it looks like. And so the PDB structures that ones I looked at a lot. All right, so when you go to a structure page, so if you type in the first front page, structure is from the experiment. There are quite a few structures containing viral new intro, what's called other. That's just a tool on the NCBI site that permits you to search rapidly. It's called basically local line search technique. We'll talk about that later in this class. It's a search against structures in their large database called matches your database of your sequence of interest. There's also another database that I use sometimes called Uniprot. There's annotations of sequences, matches and structures that are features of the protein that you might be interested in, like phosphorylation sites, signal peptides in the protein. So this is a nice site as well. And for viruses, there are actually a fair amount of database and resources out there as well. So this one here at the top is called Viper. That stands for Virus Packaging Resource, and that contains some neat little pages where you see they're devoted to specific classes of viruses, dengue, Lassa, Zika, Hep C virus. So we have a lot of things represented on there, so that you can sequences, you get things with molecular structures. This is a really nice, fairly comprehensive database resource. There's also Los Alamos National Lab. They have a pretty well-used and long-standing HIV sequence database and immunology database there too. They have one for HCV that's not really used, or not as much as it used to be. But yeah, the renal HIV database, so nice one here for HIV. Okay, so basically to summarize, there are numerous databases out there for computational biologists and also virologists specifically to refer to. Let's see, protein sequences, protein structures, of course, nucleic acid sequences as well, nucleic acid structures. So there are databases that are devoted to viruses like Viper, and also virus-specific ones like the renal HIV database. And so this is not an exhaustive list. There are many that I left off there. I noted here. There's actually a coronavirus database we've been looking at recently called TrillDB. That was developed a while ago, and of course now people do. I'm sure used a lot as well. All right, so I wanted to now frame up leading to a question with this. So if we did just downloaded a viral structure from the protein data bank, so we have the PDB page, we found a structure that was cool, we downloaded the coordinates of that structure in the PDB format. What can we do with it? How do we look at it and find out what the structure looks like? So this actually leads me to molecular visualization programs that are out there. I've listed a handful of them here, but there are of course many others. So the ones I've listed are Rasmol, Chimera, Hymol. And so Rasmol is a long sort of old school program. I'll talk about that on the next slide. Yeah, indeed, I do. Here's an old school visualization program. So it's actually something that I used 10 or 20 years ago when I was in graduate school. I believe some people still use it. It's nice to come up with a really quick representation of the structure. It's very fast. You can do it on the line. It's nice and fairly simple, but also good graphical user interface. It's available on this website. There are maybe a few minor downsides to it. It doesn't seem to representation through an old school program. I'll say a handful of them here. There are, of course, many others. So the ones that I listed are Rasmol, Chimera, and Hymol. And so Rasmol is a long sort of old school program. I'll talk about that in the slide. Yeah, indeed, I do. Here's an old school visualization program. So it's actually something that I used 10 or 20 years ago when I was in graduate school. I believe some people still use it. It's nice to come up with a really quick representation of the structure. It's very fast video on the line. It's nice, fairly simple, but also good graphical user interface. It's available on this website. There are maybe a few minor downsides to it. It doesn't seem to be very actively updated or developed. You don't really have an easy way to output high quality images if you're trying to make a paper publication with a figure for it that's showing you what the structure. Rasmol might be the best thing for that. And then it's hard to see multiple structures of it. So if you want protein A and protein B, they're similar to each other in the same viewing window in Rasmol. At least back when I was using it, that was readily achievable. Another program is called Chimera. And so this is fairly widely used. You see this reference to a lot of publications and things. And so this has lots of features. You can fit structures into EM maps, so electron microscopy will generate what they call a map. So these blobs or shapes that the molecule might fit into. And if you have a known structure that's high resolution, you can fit that into EM map in Chimera. There's also ways to do sequence alignment, sequence conservation. Backbone angle around Chandra plots, which is sort of a way to look at the angle architecture between two residues of protein backbone. You see if that particular residue fits into what's generally observed structures in terms of protein backbone. So Chimera is hosted at UCSF, University of California, San Francisco. This website. There's also a newer version out called Chimera, which is supposed to be the better graphical user interface. But the program that I use generally is called Hymol. And so this is a Python-based molecular visualization program. It supports high-resolution export structured images and movies. And so you see people using that in their publications, myself included. Look at multiple structures, Hymol, edit structures, you're looking at structures in PDB. You can go in and try to change the placement of residues or mutate or change the residue in Hymol. Hymol lets you save the session, it's not just the structure itself, if you do certain things. Try to change the placement of residues or mutate or change the residue in Hymol. And Hymol lets you save the session, it's not just the structure itself, if you do certain things.
brought this so that I can put my elbow on it because I don't want a bad back. And if I keep doing this with my elbow down, it's not going to be good for me. That should be good enough. Maybe I should just learn to keep my head up with my neck and my back instead of letting my hands hold my head up. Highland shake.
explicitly mentioned in the article, which is great. So you can go to the PDB website, you can type in those PDB codes in. All right, so this is the fourth part of the first class. So I just wanted to have a quick introduction to the exercise here. And so since we've been talking about molecular viewers and databases, but give a scenario here. So if you have a viral protein, let's call it X, you're looking to see what the structure is. Sometimes you can find that in the publication where the structure is first reported. So in the case here shown on the top, you'll see it says PDB ID codes 506A and 506B. The red box are just explicitly mentioned in the article, which is great. So you can go to the PDB website, you can type in those PDB codes in, and follow the structure. Also, you can use the PDB website and go onto there and use their search box. And if you have a particular protein, like the Zika envelope protein, you might wonder if you can find that protein in the PDB. Or some other virus, viral envelope protein, viral protease. You just type in that particular virus and a protease, and usually you'll get the structures for that if they exist in the PDB. They'll come up one of the first hits in the search. The downside of that, of course, is you might get other things too that might happen to match those words that may not be exactly what you're looking for. So you have to be a little bit more careful with that. And the exercise is related to this. And so the idea is to, of course, use your laptop, which we'll do this over Zoom, so that won't be a problem, or your desktop computer, if you have to be using that. And so the exercise will be on the course website on Elms, and so I'll put it up there. You can look at it before if you'd like. But we'll go through it over Zoom too. And the idea is to use NCBI BLAST, so I'll give a viral sequence, and use BLAST to find matches to it in the PDB. And then, the second part of that is to download the program TimeAll, and then use that to just load up and view viral protein structure. Just basic, get that working and see if that check out a structure, and we'll do more cool things with it later, of course. All right, and so this lab exercise session will be through Zoom, and we'll hold it at 5 p.m. on Monday. I hope to see you all there. Talk to you soon. One example of an asymmetric unit is here. So this shows the second part of week 1, and we'll be discussing advanced structure viewing and structural analysis. So for the first assignment for the lab exercise, just to review what we did, the main idea was to take, hello everyone, and welcome back. So this lecture is the second part of week one, and we'll be discussing advanced structure viewing and structural analysis. So for the first assignment, for the lab exercise, just to review what we did, the advanced structure viewing and structural analysis. So for the first assignment, for the lab exercise, just to review what we did, the main idea was to take a sequence that is given a sequence to was for the structural analysis. So for the first assignment, for the lab exercise, just to review what we did, the main idea was to take a sequence that is given and blast that against the protein data bank sequences. So we did that during the exercise, looked among the hits to find the closest matching protein structure, and then for the second part of that, involved installing the program primal and then loading the structure. So hopefully everybody's had the chance to do that. And if you have any problems, any questions at all, please let me know. I'm happy to talk to you and help you out. Just to review, this is from something we talked about in the previous lecture. So there's a couple of ways one can everybody's had the structure, protein data bank review. What we did, the main idea was to take a sequence that is given and blast that against the protein data bank sequences. So we did that during the exercise, looked among the hits to find the closest matching protein structure, and then for the second part of that, involved installing the program primal and then loading the structure. So hopefully everybody's had the chance to do that. And if you have any problems, any questions at all, please let me know. I'm happy to talk to you and help you out. Just to review, this is from something we talked about. Questions at all, please let me know. I'm happy to talk to you and help you out. Just to review, this is from something we talked about in the previous lecture. So there's a couple of ways one can find the structure of a protein of interest. So usually within the article that reports the protein structure, you'll see there's a data availability section or some section either at the beginning or the end of the article where they'll list the actual PDB IDs. In this case, 506A and 506B show red boxes here. And also, if one has a particular protein of interest, they know the name for it. So in this case, the SARS spike, you can enter that search in the search box on the protein data bank website. Sometimes that'll give you what you're interested in. Often you'll also find other things you might not be interested in, but if they do have that particular protein structure, that is another way to look for it. And during the exercise, we addressed another way to do it, which is basically using a blast to take, if you have a sequence of interest, you can blast against the PDB on the And so you can go in and blast the site, finding it that way. This is a more, I guess, advanced do-it-yourself method.
I'm gonna go take a break. Ugh, my back hurts. I think that was a 15 to 16-minute break. But now I'm back. But like,
if you're interested in, often you'll also find other things that you might not be interested in, but if they do have that particular protein structure, that is another way to look for it. And during the exercise, we addressed another way to do it, which is basically using BLAST to take, if you have a sequence of interest, you can BLAST against the PDB on the NCBI BLAST site, finding it that way. This is a more, advanced do-it-yourself method to do that. So one can go to the protein data bank. They have a site to download certain files. So that web page is given here at the top of the web. Go to the protein data bank. They have finding it that way. This is a more, advanced do-it-yourself method to do that. So one can go to the protein data bank. They have a site to download certain files. So that web page is given here at the top of the web. And the idea is to download. They have a large FASTA file that provides all sequences in the protein data bank. So it's a large text file. It's 130 megabytes, or at least it used to be. It's probably even more now. Since I didn't check it for at least a few months. It looks like this. It says download sequences. Shown on the right here. You can download the gzip version. That usually makes it a little bit easier. Once you get that, you can press it on your computer. And then once you get that file, a large file of protein data bank sequences, you can obtain the BLAST tool from the NCBI site. So if you just Google BLAST database with the web server, and so that's another way to do this. Another option, if you have a sequence that you would like to see if there's any corresponding structure for it in PDB. Next, I want to talk about what is in a PDB file. So during the exercise and also during the previous lecture, we talked about these PDB files that represent structures of molecules. And there's several parts to the PDB file. The first part of the PDB file that when you download, you can look at it. It's basically like a text file. The first section of it is header lines. So you'll see that that's shown on the right here in this slide. So it says certain basic things like the deposition date, the PDB code. So this one was September 24, 2013. The PDB code is for NWF. Title, authors, other things like that, and header lines, experimental details, what the different proteins are. So that's all within the header lines. And then once you scroll down a little bit farther into the file, past the header lines and other descriptive regions of the PDB file, you get to the app. Next, I want to talk about what is in a PDB file. So during the exercise and also during the previous lecture, we talked about these PDB files that represent structures of molecules. And there's several parts to the PDB file. The first part of the PDB file that when you download, you can look at it. It's basically like a text file. The first section of it is header lines. So you'll see that that's shown on the right here in this slide. So it says certain basic things like the deposition date, the PDB code. So this one was September 24, 2013. The PDB code is for NWF. Title, authors, other things like that, and header lines, experimental details, what the different proteins are. So that's all within the header lines. And then once you scroll down a little bit farther into the file, past the header lines and other descriptive regions of the PDB file, you get to the atom lines. And so these contain the actual coordinates of the protein or molecule that you're looking at. And so the coordinates are encoded by lines that either start with atom, ATOM, all caps, or head atom. And atom is usually for typical protein residues, head atoms, small molecule ligands, other things. So on an atom line, you'll see the coordinate given, so the x, y, and z coordinates, units and angstroms, and other identifiers for each atom line. So each atom line represents one atom in the molecule. So the top line here is a carbon atom, GOY, that means it's a glycine residue. It's residue number 530, and then so on, it has the x, y, and z coordinates. Basically goes, and each line represents a different atom on this particular molecule. This is all chain C here, this file. And as I mentioned, so these typical amino acids are called atom, and then the head atom lines are these lines stressed like that. That means it's a small molecule or ligand. There are other things that are in an atom line, something called the V factor. So V factor is basically a representation of the residue mobility as the x, y, and z coordinates. Basically goes, and each line represents a different atom on this particular molecule. That's the descriptive regions of the PDB file. You get to the atom lines. And so these contain the actual coordinates of the protein or molecule that you're looking at. And so the coordinates are encoded by the lines that either start with atom, ATOM, all caps, or head atom. And atom is usually for typical protein residues, head atoms, small molecule ligands, other things. So on an atom line, you'll see the coordinate given, so the x, y, and z coordinates, units and angstroms, and other identifiers for each atom line. So each atom line represents one atom in the molecule. So the top line here is a carbon atom, GOY, that means it's a glycine residue. It's residue number 530, and then so on, it has the x, y, and z coordinates. Basically goes, and each line represents a different atom on this particular molecule. This is all chain C here, this file. And as I mentioned, so these typical amino acids are called atom, and the head atom lines are these lines stressed like that. That means it's a small molecule or ligand. There are other things that are in an atom line, something called the B factor. So the B factor is basically a representation of the residue mobility based on the X-ray diffraction data. It gives you an idea of how much movement or vibration there is within the crystal for this particular atom. And you can see that on the right here on this slide. Column is red here, B factor. So the first number here is 58.13 for that first atom, and so on down through the rest of the atoms. And if you look and in a PDB file, there's a pretty big range of B factors. In PyMol or other molecular visualization programs, there are options to color residues and atoms by B factors. So that's shown here on the lower left. For this particular structure, this is actually the same structure that we have some atoms for over on the right here. The full structure is shown on the left. So higher B factor means a more vibration, more mobility. And you can see those red corresponding to high B factor here, sort of like surface residues and things in certain regions, whereas some other regions are more green and bluer colors, and that represents over basically fixed regions of the protein. That's pretty cool way to visualize interesting feature of molecular structures. Another part of a PDB file is
No, I'm just trying to study, just to keep up. I have an online class, and I hadn't watched uh, the last two sessions of the class, so I'm watching them and then applying it to the the program that we're supposed to be using. Because I'm gonna have to make a project with this, so and it's kind of cool. Right now I'm looking at the structure of one of the structures of a virus that's related to the viruses I work with. Can you
Asymmetric unit.
I think it's because everyone's on it.
So usually by default, if you have a PDB file, you're given the asymmetric unit. So when they solve the crystal structure, there might be two different forms of the protein molecule that are slightly different from one another. They repeat themselves in the crystal lattice. And that's a pretty cool way to visualize interesting feature of molecular structures. Another part of a PDB file is the asymmetric unit. So usually by default, if you have a PDB file, you're given the asymmetric unit. So when they solve the crystal structure, there might be to
That's it, right? Oh no! Ice, baby.
different forms of the protein molecule that are slightly different PDB file is the asymmetric unit. So usually residues and atoms by B factors. And so that's shown here on the lower left. For this particular structure, this is actually the same structure that we have some atoms for over on the right here. The full structure is shown on the left. And, so higher B factor means more vibration and mobility. And you can see those red correspond to high B factor here, sort of like surface residues and things in certain regions, whereas some other regions are more green and bluer colors, and that represents more basically fixed regions of the protein. That's a pretty cool way to visualize interesting feature of molecular structures. Another part of a PDB file is the asymmetric unit. So usually by default, if you have a PDB file, you're given the asymmetric unit. So in this solid crystal structure, there might be two different forms of the protein molecule that are slightly different from one another that repeat themselves in the crystal lattice. And that will mean that when you download the file, if you're looking at getting a particular structure for one protein, you might actually have a couple of copies of it that represent two slightly different forms of that in terms of their structures. Different from one another, they repeat themselves in the crystal lattice. And that will mean that when you download the file, if you're looking at getting a particular structure for one protein, you might actually have a couple of copies of it that represent two slightly different forms of that in terms of their structures. And so you can, later I'll talk about ways to, so you can just get one form of the molecule. But in some cases, too, in the asymmetric unit, you'll just get one copy of what's actually part of a larger assembly, like a biocapsid. And so that's another thing that you can have to consider when you download the PDB file. One example of an asymmetric unit is here. So this shows the protein data bank file that code is for MWF. So this is a hepatitis C virus E2. Some envelope lipoprotein complex with the AR3C antibody. This has two copies in the asymmetric unit of the antibody and the E2 protein. And so up on top here is one copy, on the bottom, the magenta, very, very pink is the other copy of that complex. So there's basically a crystallographic interface where you see these are the actual interfaces between the antibody and antigen, and that's the way they sit together in the crystal lattice, as indicated here, in between the yellow E2 antibody. So the two interfaces with the actual binding interface between the antibody and the antigen, but also a crystallographic interface. So the question is, how do you just get one complex from that asymmetric unit? And so, as I mentioned, there's ways to do that. Within Primal, you can set tell_all through this command set assembly, (1). And that will give you first what they call bioassembly, that tell_time complex from that asymmetric unit. And so, as I mentioned, there's ways to do that. Within Pymol, you can set tell_all through this command set assembly, the actual binding interface between the antibody and the antigen, but also a crystallographic interface. So the question is, how do you just get one complex from that asymmetric unit? And so, as I mentioned, there's ways to do that. Within Pymol, you can set tell_all through this command set assembly, (1). That will give you first what they call bioassembly, that molecular structure, in this case a complex. So when you say fetch or MWF, that will get that PDB structure, and since you said set assembly 1, that will actually get that only the one assembly rather than the full asymmetric unit, which will have two copies of it. Another example, as I mentioned, sometimes you see viral capsid proteins, and they only give you one copy, only a part of. Another example, as I mentioned, sometimes you see viral capsid proteins, only the one assembly rather than the full asymmetric unit, which will have two copies of it. Another example, as I mentioned, sometimes you get that only the one assembly rather than the full asymmetric unit, which will have two copies of it. Another example, as I mentioned, sometimes you see viral capsid proteins, and they only give you one copy, one little part of the capsid, which gets repeated symmetrically to generate the entire capsid surface. It's molecularly relevant protein. In this case, you say set assembly 1, once again, and then fetch, in this case, 1 IHM. And that's the structure of the norovirus capsid. This is for norovirus. Do some other things to make it look interesting. So, show cartoon, color by chain, and then background in white. So that generates the picture like that. This. So you see the full capsid, as you can see, there are repeats of magenta and green proteins across the capsid surface. And so you get the full, what they call bioassembly, this capsid protein, which is pretty cool. Right, one other way to get the bioassembly is directly through the PDB site. So if you search one IHM on the PDB front page, that will bring you to the page for that structure. So you download files in biological assembly 1 from that page of that structure. You get the PDB format, bioassembly 1. You'll get just the 3 subunits of the asymmetric units, so you need to actually specify a biological assembly 1. And then when you get that downloaded file and unzip it, you can open it in Primal and you get the full, you said download biological assembly 1. This is another way to directly get that file versus going through Primal. That will give you the full capsid in this case. All right, so in this case, when you download the structure and you load it into Primal, it will still only show the asymmetric unit, even though the full biological assembly is actually present in the PDB file. So in this case, you have to say select movie in the Primal window and then show all states. And basically, because the different copies of the biological assembly of the molecule of the assembly are not shown by default. They're sort of encoded as different states in the movie. When you one example of an asymmetric unit is here. So this shows the protein data bank file that code is for MWF. So this is hepatitis C virus B2. It's an envelope glycoprotein complex with the AR3C antibody. This has 2 copies in the asymmetric unit of the antibody and the E2 protein. And so up on top here is 1 copy, and on the bottom the magenta and the gray and pink is the other copy of that complex. So there's basically a crystallographic interface where you see these aren't the actual interfaces between the antibody and antigen, but that's the way they sit together in the crystal lattice, as indicated here in between the yellow E2 and the antibody. So there's interfaces both the actual binding interface between the antibody and the antigen, but also a crystallographic interface. So the question is, how do you just get 1 complex from that asymmetric unit? And so, as I mentioned, there's ways to do that. Within Primal, you can set tell Primal through this command set assembly, comma 1. That will give you first we call bioassembly, that molecular structure, in this case a complex. So we say fetch or MWF. That will get that PDB structure. And since you said set assembly 1, that will actually get that only the one assembly rather than full asymmetric unit or 2 copies of it. Another example, as I mentioned, sometimes you see viral capsid proteins, and they only give you 1 copy, one little part of the capsid, which gets repeated symmetrically to generate the entire capsid surface. It's molecularly relevant protein. In this case, you say set assembly 1, once again, and we fetched, in this case, 1 IHM, and that's the structure of the normal virus capsid. This is from virus. Do some other things to make it look interesting. So show cartoon, color by chain, and then background white. And so that generates the picture like this. So you see the full capsid. As you can see, there are repeats of magenta and green proteins across the capsid surface. And so you get the full, they call bioassembly, this capsid protein, which is pretty cool. One other way to get the bioassembly is directly through the PDB site. So if you search 1 IHM on the assembly column 1, that will give you first what we call bioassembly, that molecular structure, in this case a complex. So we can say fetch or MWF. That will get that PDB structure, and since you said set assembly 1, that will actually get that only the one assembly rather than full asymmetric unit, which will have 2 copies of it. Another example, as I mentioned, sometimes you see viral capsid proteins, and they only give you 1 copy, one little part of the capsid, which gets repeated symmetrically to generate the entire capsid surface. It's molecularly relevant protein. In this case, you say set assembly 1, once again, and we fetched, in this case, 1 IHM, and that's the structure of the virus capsid. So this is from virus. Do some other things to make it look interesting. So this is show cartoon, color by chain, and then background white. And so that generates the picture like this. So you see the full capsid, and as you can see, there are repeats of magenta and green proteins across the capsid surface. And so you get the full, they call bioassembly, this capsid protein, which is pretty cool. One other way to get the bioassembly is directly through the PDB site. So if you search 1 IHM on the PDB front page, that will bring you to the page for that structure. And you can say download files. Another example, as I mentioned, sometimes you see viral capsid proteins, and they only give you 1 copy, one little part of the capsid, which gets repeated symmetrically to generate the entire capsid surface. It's molecularly relevant protein. In this case, you say set assembly 1, once again, and we fetched, in this case, 1 IHM, and that's the structure of the normal virus capsid. So this is from virus. Do some other things to make it look interesting. So show cartoon, color by chain, and then background white. And so that generates the picture like this. So you see the full capsid. As you can see, there are repeats of magenta and green proteins across the capsid surface. And so you get the full, what they call bioassembly, this capsid protein, which is pretty cool. One other way to get the bioassembly is directly through the PDB site. So if you search 1 IHM on the PDB front page, that will bring you to the page for that structure. And then you can say download files and then biological assembly 1 from that page of that structure. And you get the PDB format, bioassembly 1, little just the three subunits of the asymmetric unit, so you need to actually specify biological assembly 1. And when you get that downloaded file and unzip it, you can open it in Primal, you'll get the full, you said download biological assembly 1. This is another way to directly get that file versus doing it through Primal. That will give you the full capsid in this case. All right, so in this case, when you download the structure and you load it into Primal, it will still only show the asymmetric unit, even though the full biological assembly is actually present in the PDB file. So in this case, you have to say select movie in the Primal window and then show all states. And basically, because the different copies of the biological assembly of the molecule within the assembly are not shown by default. They're sort of encoded as different states in the movie. When we say movie show all states, it will show the rest of the structure. And then you can, once again, color by chain, show ribbons to make it look sort of like you see what we're doing right here. Hello, everyone, and welcome back. So this lecture is the second part of week 1, and we'll be discussing advanced. Another example, as I mentioned, sometimes you see viral capsid proteins, and they only give you one copy, one part of the capsid, which gets repeated symmetrically to generate the entire capsid surface. That's molecularly relevant protein. In this case, you say set assembly 1. Once again, and we fetched, in this case, 1 IHM, and that's the structure of the virus capsid. So this is for virus. One other way to get the bioassembly is directly through the PDB site. So if you search 1 IHM on the PDB front page, that will bring you to the page for that structure. You can say download files and biological assembly 1 from that page of that structure. And you get the PDB format, bioassembly 1. You'll get just the 3 subunits of the asymmetric units, so you need to actually specify a biological assembly 1. And when you get that downloaded file and unzip it, you can open it in Primal and you get the full, you said download biological assembly 1. This is another way to directly get that file versus going through Primal. That will give you the full capsid in this case. All right, so in this case, when you download the structure and you load it into Primal, it will still only show the asymmetric unit, even though the full biological assembly is actually present in the PDB file. So in this case, you have to say select the one you see on the right here. For this part of the lecture, we'll talk about structure alignment, as well as a few more commands I will program. One thing people often need to do when they are looking at molecular structures, especially if they find multiple ones, maybe different antibodies that are bound to the same target. For this part of the lecture, we'll talk about structure. One thing people often need to do when they are looking at molecular structures, especially if they find multiple ones, maybe different antibodies that are bound to the same target, different forms of the same protein that might have different conformations, is they need to superpose them. So they put them into the same reference frame. And so this can be done both for experimentally determined structures as well as structural models. If you have different docking models, for instance, we'll talk about protein-protein docking later. And if people have different models of a ligand, for instance, docked to a receptor protein, you might want to compare to see how similar those different models are to each other. So one might need to superpose the different models to put them in the same reference frame. Especially experimentally determined structures, different groups will use just arbitrary X, Y, Z coordinates as the origin and orientation of proteins as well. So it's necessary to superpose different structures. In this example here, I just wanted to show orientation of proteins as well. So it's necessary to superpose different structures. In this number, for instance, docked to a receptor protein, you might want to compare to see how similar those different models are to each other. So one might need to superpose the different models to put them in the same reference frame. Especially experimentally determined structures, different groups will use just arbitrary X, Y, Z coordinates as the origin for the orientation of proteins as well. So it's necessary to superpose different structures. In this example here, I just want to show what we can do when we superpose structures. So this is showing a lot of different T-cell receptor structures that are bound to peptide and MHC molecules. So T-cell receptors are sort of like antibodies, or immunoglobulin proteins that are used by the immune system on the surface of T cells to recognize peptide antigens that are presented by the major histolytic complex or MHC. And so we collected in my lab a whole lot of these different structures, over 130 of them. And so the MHC structures are at the bottom of this figure here. So this final related that was shown, just a snapshot of it. So that's the Simon's cartoon. We superposed all these different complexes according to the MHC molecule. Sort of put them all on top of one another. We can see using this that there's a pretty big diversity in terms of where the T-cell receptors sit. People know that they all bind the same general binding site. There's actually a pretty big level of diversity within the range of what's experimentally seen for T-cell receptors. So this is a kind of cool way to visualize that and see how these things are different from one another. If you just download two different T-cell receptor peptide MHC complexes from the PDB, they might be all over the place next to each other or anything like that. So it's necessary to superpose them like this to get a better handle on how similar they are to each other. Another example I'm going to talk about is the hepatitis C virus area. So there are a couple of antibodies that were described fairly recently by a group, Caltech, in collaboration with the researcher at Johns Hopkins, Vanderbilt. And so these antibodies are called Hep C3 and Hep C74. They're both identified from patients that were infected with hepatitis C virus. So these are human antibodies. They target the ACVE2 envelope glycoprotein, which just like that AO3C we talked about recently. Fairly recently, a group Caltech, in collaboration with the researcher at Johns Hopkins and some other folks at Vanderbilt. And so these antibodies are called Hep C3 and Hep C74. They're both identified from patients that were infected with hepatitis C virus. So these are human antibodies. They target the ACVE2 envelope glycoprotein, just like that AR3C antibody we talked about recently. So that we have X-ray structures that were described in this article here, antibody A2 complexes. The question is, can we compare these and just see whether these actually have the same sort of binding site on the A2 protein, if they bind in different angles, different mechanisms to target this protein. This is from that paper where they talk about the different structures. So the PDB codes are at the top here. I think I put them in myself. So the 6MEI, 6MEH, and 4MWF. So these are all experimentally determined structures, these three different antibodies. And you can sort of see, even in this representation here from that paper, that there is some similarity between Hep C3 on the left and Hep C74 in the shapes, the way the angles these antibodies come in on the bottom, roughly the same, whereas 4MWF seems to be different. But the question is, can we look at that ourselves? And so that's what we use structural alignment for. And if we're trying to align two different structures, we can't always assume that they have the same number of residues or lengths. And so it is necessary when you're performing an alignment between proteins or other types of molecules to identify an alignment that will optimally be able to compare the two different proteins. So in this case, we have protein A and protein B on the right in the figure, and we want to see what residue correspondence would allow a good fit between these two molecules. So structural alignment programs will be able to identify basically an alignment. So you see in the log here, protein A, it puts gaps in here. Protein B, there's no gaps, but protein A needed the gaps in order to successfully align with protein B. And so you usually look for equivalent residues like alpha carbon atoms on the backbone. And so you get this gap alignment. And there are some other methods that are a little more advanced. They don't even care about the order of the residues or alpha carbons in the sequence. And they'll use geometric-based methods. But either way, they find some correspondence between residues in the two structures. If they don't have the same lengths, to find ways to line those up. And you can get superposition once you have a sort of alignment between residues. There are lots of structural alignment tools available with this website called proteopedia.org. You can sign up and check it out if you want. A list of structural alignment servers that are available. So these are web servers or websites. Use those to align structures or in some cases to circle. A given structure against the protein data banks, large set of structures that are experimentally determined. And this is, of course, not a full list. This is a big list already. So there's a lot of things out there that have been developed over the years to address this challenge. So one program that I mentioned a couple slides ago, there's also on the previous slide, it's called Dolly. So this is a nice program that can both perform pairwise alignments between proteins and also search against the entire protein data bank. So if you have, for instance, this HIV and below protein, this truncated part of that, and so this is a viral protein structure that you found, you want to see if there's any other structures that are known, whether they be from a virus or maybe something else that match this structure for HIV. And so you would enter that into the Dolly interface. And even if the sequences are nowhere near the same, if there happens to be structural matches to that protein fold, then in this case, HIV envelope and magenta, Dolly 1 with that give a rank listed on the score, which represents the level of structural alignment with your input protein. So this is a nice web server. It generally takes a few minutes to maybe an hour or so to search the entire PDB with your query protein and how busy they are. It's a nice program and server to be aware of. So if you have two different structures or a few different structures, visualize those in program Primal, you can also use Primal itself to align those structures and put them in the same reference frame. So these are the structures that I was talking about earlier that represent those antibodies that were described in that article. And so FC3 and FC74. And so we can fetch those two structures. So fetch 6MEI, fetch 6MEH. And then once we clean up the structures, hide things like water molecules, other things you don't necessarily want to look at in the window, and you can show cartoon. So that shows just the cartoon representation of the proteins, and then color by chain, and that will allow you to see the envelope protein and the antibodies colored separately, which is pretty useful. But you can see here that these two different structures, even though they're close to each other, they're not too far in terms of Cartesian coordinates, they're not in the same reference frame, so the antibodies and the antigens are sort of flipped with respect to one another. In this case, you might want to see how the antibodies sit if you can superpose the antigens into the same reference frame. So that would be the green chains, the E2 portions. So if you look at or try to use a program like Primal and just tell it to superimpose 6MEH onto 6MEI, it won't necessarily do what you want if you just want to superimpose a specific portion onto another portion of the other structure. So we want to just superpose the E2 portions, just the viral part. You can see that in one case it's chain C. So we make a selection here. You say select E2, so that's the name of the selection. Then chain C and 6MEI, so that will select this left-hand green chain here. Make a selection called E2, which represents all the residues in that chain. And what we want to do here is to align the 6MEH to that particular E2 ion, so that will select, make a selection here, say select E2, so that's the name of the selection, then chain C and 6MEI, so that will select this left-hand green chain here, make a selection called E2, which represents all the residues in that chain. And what we want to do here is to align the 6MEH to that particular E2, and so that they'll be in the same reference frame. And so then we perform 6MEH, click on the A part, and align to selection, and then E2, which is that selection that we just made. That will superpose the 6MEH structure onto just the E2 part. So we'll take the E2 from 6MEH and basically superimpose that onto the E2 part of 6MEI. In this case, you can see that there's a pretty good correspondence between the structures, and this is after the superposition shown in the window here. Primal will also report the root mean square distance between the structures, so in this case it's just about 26 angstroms, so that's really close to each other. It's basically a very good match in terms of that. E2 parts, but also even the way the antibodies sit as well. And so Primal won't necessarily try to line every single residue to each other. It will skip residues. Just as I mentioned, sometimes it's structural alignments and they'll have gaps and leave out residues. Primal will, using its own algorithm, decide residues to scrap from the alignment. So you do have to be a little bit aware of that sometimes. There's also a super command that I think performs basically the same thing, where you can tell it to superpose one structure onto another. In this case, on the bottom here, based on the CA or alpha carbon atoms. That's another method to use, but I generally use the approach that I described here by making the selections. And then 690HA aligned to selection approach. Another cool thing you can do in Primal is to measure distances between atoms. So in this case, you're looking at a structure and the antibody bound to, once again, HCBE2. In this case, it's just an epitope, it's a small region of HCBE2. So residues 412 to 423 on this PB, which is the code is 4GGY. We're interested, in this case, in looking, this example, at residue 415, because we know from there actually being clinical studies with this antibody, putting that in patients, and there have been some reports of this virus escaping from the antibody based on mutations at residue 415. The question is, in this structure here, can we look at residue 415 and determine whether it's actually close enough to the antibody to say that this is probably it's mutations of that residue, since it's so close to the antibody, is probably the reason why it's escaping. It's pushing away the antibody or somehow interacting with it in a favorable way. All right, so when we go into the primal window, once you have this structure up, in this case 4DGY, shown on the right here, you can go into wizard at the top, and measurement and distances. So that will have this show up here, this measurement window. You can click on distances. And, so one more thing I want to note here is about selections in Primal. So you can click on distances and distances window. Once you have the new antibody, it's prompted to terminate studies with this animal. We're interested, in this case, in looking at this example at residue 415, because we know from there are actually clinical studies with this antibody, including that in patients, and some reports of this virus escaping from the antibody based on mutations at residue 415. The question is, in this structure here, can we look at residue 415 and determine whether it's actually close enough to the antibody to say that this is probably it's mutations of that residue. Since it's so close to the antibody, it's probably the reason why it's escaping. It's pushing away the antibody or somehow interacting in an unfavorable way. All right, so when we go into the primal window, once you have this structure up, in this case, it would be GY, shown on the right here, you can go into wizard at the top, and measurement and distances. So that will have this show up here, this measurement window. And you can click on distances. And so one more thing I want to note here is about selections. And go up here, this measurement window, wizard at the top, and measurement and distances. So that will have this show up here, this measurement window. And you can click at the top, and measurement and distances. So that will have this show up here, this measurement window. And you can click on distances. And, so one more thing I want to note here is about selections in Primal. So you can select, as I noted, we can select chains. We can also select specific residues to highlight in addition to just chains. So or other things as well, like certain atoms by name. So basically the syntax of that is just saying select you want stuff, or U2, or DP, or arbitrary you want. And then the selection criteria. So in this case, instead of the chain, we're going to call it, well, the name of the selection will be TripRes, TRP, RDS. And then we're saying residue 420, residue 420, and chain A. So this is on chain A, residue number. It's mutations in that residue. Since it's so close to the antibody, it's probably the reason why it's escaping. It's pushing away the antibody or somehow interacting in an unfavorable way. All right, so when we go into the primal window, once you have the structure up, in this case it would be GY, shown on the right here, you can go into wizard at the top, and measurement and distances. So that will have this show up here, this measurement window. You can click on distances. So one more thing I want to note here is about selections in Primal. So you can select, as I noted, we can select chains, but we can also select specific residues to highlight in addition to just chains. So there are other things as well, like certain atoms by name. So basically the syntax of that is just saying select when you give it any name you want, call it stuff, call it YouTube, call it VP, or any rubber string you want. And then the selection criteria. So in this case, instead of the chain, we're going to call it, well, the name of the selection will be TripRes, TRP, RES. And then we're saying residue 420, residue 420, and chain A. So this is on chain A, residue number 420. And in this case, this is residue 420 here, shown on the right. And we've colored it red, shown it sticks. You can do that through the graphical user interface for that selection. Once you've selected it, you can see, choose to color it or represent it in a certain way. You can also go ahead and click on residues in the Primal window, and that will also select residues by default. It calls it SE, and you'll be able to do things with that residue if you'd like through that as well. You just click on them. You can also specify residues, as shown here by residue number and chain, atom name, other things you want to select by. All right, so once you've gotten the residue selection, things highlighted, and if you're interested in those particular ones, in this case, we want to see, as I mentioned, residue 415 on chain A. As I mentioned, it's an asparagine. When we go through that measurement window, we can see the measurement distances. You click on these two atoms. In one case, the N415. In this case, there's a tryptophan on the antibody. Primal will report the distance between them. In this case, it's really close. It's 3.8 angstroms. These are pretty much right in contact. And this asparagine, if you look at the structure, you'll also see it's pretty varied in the antibody. So there's really no surprise that mutations on that asparagine as we do reach a viral speed. All right, so just to summarize, I just want to talk about mutations on that asparagine as we do reach a viral speed. All right, so just to summarize, I just want to talk about things we've gone over during this class today. So we can search for structures of viral proteins through PDB, also using BLAST. Visualizing the molecules in Primal, there's ways to do things like align structures, calculate distances between the atoms. And even though we've sort of summarized a couple of different things, I wanted to point you to a couple of useful resources. One is primalwiki.org. So this has a pretty good set of documentation. can also select residues by default, called SE. Be able to do things with that residue if you like through that as well. You just click on it. You can also specify residues, as shown here by residue, number, chain, atom name, other things you want to select by. All right, so once you've gotten a residue selection, things highlighted, and if you're interested in those particular ones, in this case, we want to see, as I mentioned, residue 415 on chain A. As I mentioned, that's an asparagine. When we go through that measurement window, we can see the measurement of distances. Click on these two atoms. In one case, the N415. In this case, there's a triplicate on the antibody. Primol will report the distance between them. In this case, it's really close. It's 3.8 angstroms. These are pretty much right in contact. And this asparagine, if you look at the structure, you'll also see it's pretty buried by the antibody. So there's really no surprise that mutations on that asparagine residue do lead to viral escape. All right, so just to summarize, I just want to talk about things we've gone over during this class today. So we can search for structures of viral proteins, PDB, also using BLAST. Visualizing the molecules in Primol is ways to do things like align structures, calculate distances between atoms. And even though we've sort of summarized a couple of different things, I wanted to point you to a couple of useful resources. One is primolwiki.org. So this has a pretty good set of documentation on selections, coloring, doing all sorts of things like movies. And we'll, as we go through the class, we'll have a chance to go through more things, especially during the exercises. But I wanted to point you to some useful references that are available online. In addition to this website, I also wanted to point you to this Primol reference card. So there's a PDF file, and I've uploaded it to the Elms Primol folder in the files. And this is sort of a fairly big set of commands, organized pretty well, so you can just read through that and do a few useful commands and some basic information about things you can do in Primol. All right, so once you've gotten a residue selection, finger, number, chain, atom name, other things you want to select by. All right, so once you've gotten a residue selection, things highlighted, and if you're interested in those particular ones, in this case, we wanted to see, as I mentioned, residue 415 on chain A. As I mentioned, that's an asparagine. When we go through that measurement window, show frequency to measurement of distances, click on these two atoms. In one case, the N415. In this case, there's a tryptophan on the antibody. Primal will report the distance between them. In this case, it's really close. It's 3.8 angstroms. These are pretty much right in contact. And this asparagine, if you look at the structure, you'll also see it's pretty buried by the antibody. So it's really no surprise that mutations on that asparagine as we do reach viral escape. All right, so just to summarize, I just want to talk about things we've gone over during this class today. So we can search for structures of viral proteins, PUB, also using BLAST. Visualizing the molecules in Primal as ways to do things like align structures, calculate distances between atoms. And even though we've sort of summarized a couple of different things, I wanted to point you to a couple of useful resources. One is primalwiki.org. So this has a pretty good set of documentation on selections, coloring, doing all sorts of things. You can make movies. And we'll as we go through the class, we'll have the chance to go through more things, especially during the exercises. But I wanted to point you to some useful references that are available online. In addition to this website, I also wanted to point you to Primal reference card. So there's a PDF file, and I've uploaded it to the Elms Primal folder in the files. And this is sort of a fairly big set of commands. They're organized pretty well, so you can read through that. And that gives you a few useful commands, some basic information about things you can do with Primal. For this part of the lecture, it's Primal reference card. So there's a PDF file, and I've uploaded it to the Elms Primal folder in the files. And this is sort of a fairly big set of commands. They're organized pretty well, so you can read through that, and that gives you a few useful commands with some basic information about things you can do with Primal.
Alright, I'm gonna take a break. I think I can take a break for food. It's 1 currently. So auf Wiedersehen. High five, Jacob. Bye.